everyone, Amy Nunnally here, and today I want to talk about the microscopic anatomy of a muscle. We talked in the last video about the gross anatomy of the muscle, and just to review that really quick, remember we have a skeletal muscle that is attached to the bone through the tendon. Alright, so each muscle is made up of multiple fascicles. So let me write that word in here. So this bundle of muscle fibers here is a fascicle. That's just the name for that. Each fascicle is a bundle of muscle fibers or cells. So here's a picture of an individual muscle fiber or cell. And that's as far as we got last time. But we need to keep going and see what's inside of this cell. So this individual cell is composed of multiple organelles. These big organelles look a whole lot like what we've seen in all these other cross sections. So you've got to make sure you don't get those confused. But each of these large organelles is called a myofibril. And these myofibrils fill up a majority of the cell. That's what packs the cell so full, and that's what actually makes the nuclei get pushed off to the edges of these massive cells. So these myofibrils are actually made up of sarcomeres joined end to end. They are cylindrical structures, okay? But in a sense, they're joined to each other almost like train cars would be, right? So end to end to end would be all these sarcomeres. But we can see that this is actually a cylindrical or three-dimensional structure. Okay, so the myofibrils are made up of two types of filaments called thin and thick filaments. These thin and thick filaments are what make up the sarcomere. And a sarcomere is the functional unit of a skeletal muscle. That means that if the function of a skeletal muscle is to contract, then the sarcomere itself will retain that ability to contract. And so all the sarcomeres working together are what contract all at the same time to make the skeletal muscle as a whole contract. So we need to look a little bit more in detail about this sarcomere and what it looks like. Here is a picture of it that we can just cover briefly, and then I'll move over to another slide. Let me erase all of that and pull out my highlighter. So each sarcomere is going to be made of, let's see, I'm going to say the thick filaments, which are made mostly of the myosin protein. These thick filaments are right here. They run down the middle of the sarcomere. You can tell they are larger than the blue filaments next to it because they contain a lot more proteins. We also have these thin filaments here in blue. These thin filaments are staggered on either side, and they go in between the thick filaments just a little bit. There are a couple of other terms here that we can talk about as well. The Z-disc, whoops, I didn't change to my pen. The Z-disc is basically going to form the edges or the boundaries of each sarcomere. You can see it here. It actually has this zigzag appearance here and here. So from one Z-disc to another is one sarcomere. Here in this diagram they're drawn as straight lines. In class, or in this class we might draw it more like this with that zigzag line to look more like it does here in real life. I'm going to go on to the next slide so we can show you this a little bit better. So here we are again with the Z-disc that runs and makes the border, the edges of that sarcomere. So here's the left-hand border and here's the right-hand border. We have in the middle these red bands are what are the thick filaments in this figure, made of, sorry, the thick filaments are myosin proteins, and the thin filaments are in blue in this diagram. Those are these smaller ones here. Those are the thin filaments. When we take a look at the sarcomere, we also see a couple of extra borders that I want to point out to you. One is that if you look at what's called the A-band goes from the edge, the outer edge of all of the thick filaments. That forms the A-band. Between all the thick filaments, where you have the Z-disc in the middle and you only have thin filaments, that makes up your I-band. So what you might notice is that you have a thick region made of the A-band, and then you have a thin region called the I-band, and another thick region, A-band, I-band, thin region. And what you can tell is that this is where the striations actually come from in skeletal muscle. Remember, those are the stripes that skeletal muscle is known for? Well, that's because of the arrangement of the thick and the thin filaments in the sarcomeres. The Z-disc, we said, was this zigzag border in the middle of the I-band that formed the junction between two sarcomeres. And then the last little piece that I need to show you and label really quick is this one called the H-zone, or the H-band. That is simply this area of gap. It's in the middle of the A-band, so we know that that's where we have our thick filaments. But in this central core, this is between the ends of the thin filaments. So the thin filaments don't come together in the middle. There's a gap in between. And so that H-zone is going to be a little bit of a lighter colored stripe right in the middle of the A-band. So this is, just to specify for you so you know for the future, this is a relaxed sarcomere. When sarcomeres contract, this structure is going to change a bit, and we'll talk about that in the next video. Good job!